should be enough. Okay, I wish to start with this picture because when we talk about libraries, usually a library then finally is a small tube with a couple of hundreds of microliters, but this is, it's already over. Nice. Yeah. So, but at the beginning, I mean, you have to prepare quite a lot of material. So we prepared 160 of these large plates, full of colonies, and this gives the dimension of the library. We'll come back later on, discuss a little bit what we mean when we talk about library diversity, dimension, and so on. But this is indeed the first library we prepared in 2005, 2006, and it's the first example of naive library for nanobodies. Yeah. Because at that time, nanobodies were extremely exotic, so were not so popular as they are now, and people thought that it was not possible to have a pre-immune collection of nanobodies, but that you have to pass any time through immunization of an animal. This is feasible, of course, but it costs time, it costs money, and effort for preparing any time a new library. So we said, okay, let's see if we can just make a collection and then use this collection. Yeah, and okay, we demonstrated it's an alternative. Then, in the more recent years, people moved from the idea of naive libraries, so made with nature material for animals, to synthetic libraries because of the cost of the synthesis decreased a lot. Yeah, so made possible this. But if you consider the evolution of the idea of getting ligands to interfere, recognize, block, stimulate, anyway, a molecular, biological target, yeah, we see that nanobodies were not the only kind of fragment.
Did you just want to check?
Better? Thank you. Yeah, nanobody has not been the only molecule proposed. Yeah, so there are DARPins, there are affibodies, monobodies, so different kind of molecules. Yeah, and when we have a look, yeah, at the yeah, it doesn't work. The mic. It's only for the camera. I can use the microphone or no? Yeah, better now?
Yeah.
Okay, sorry. Yeah, so people try different molecules with different characteristics. Yeah, so here it's indicated the major one, but indeed I calculated that. Oh, thanks a lot. That probably there are roughly 50 different scaffolds that have been proposed over the years. And in many cases, we have no idea if these scaffolds are really good or bad or better than what we use so far, because simply they have been developed in one single lab and then they died more or less there. Yeah? So only few of them managed to go outside from the original lab. So indeed, maybe there is a sort of treasure somehow hidden, and we don't know about it. Yeah. But what is important to see is they are very different in terms of form, so geometry. Yeah. So this is a classical IgG, and what is meaningful in this, yeah, it's the combination of the two variable regions of the light and heavy chains. Yeah? This is not in scale because the VH and NR correspond to one of these domains. Yeah? But exactly, I mean, people identified nanobodies from sharps and camelides, yeah? But then they identified all other scaffolds that could be useful. Yeah? And as you see, because of the different geometry on the surface, you can try to use different strategy for using these different molecules. So we can start from what is the paratops, so the binding surface of a conventional antibodies. Yeah? So it's formed by the combination of the loops, or the three loops corresponding to the CDR 1, 2, and 3 of the light and the heavy chain. So they form altogether a combination of 6 components, yeah, that in most of the cases, yeah, they form a sort of bowl, yeah, as indicated here, recognize extruding surfaces on the antigen or flat surfaces on the antigen. So this is the classical way in which conventional antibodies can recognize the antigen. So it means that somehow they discriminate on the surface of the antigen just some surfaces, some epitopes. Yeah? So it means that it's difficult to target with this kind of antigen some specific epitopes on the antigen. And it is known. So it's known that the conventional antibodies have troubles to block some specific epitopes, for instance, hidden epitopes in viruses and so on, because they cannot reach it. Yeah. So this is already one reason for which you think that just sticking to one single ligand, yeah, could be not optimal, but you must have more options. Why? Well, consider how work the nanobody. The nanobodies is a single domain molecule, so it means that with respect to a conventional antibody, it's missing 3 loops. So it means that in theory, yeah, it should have a shorter or smaller surface of interaction with the antigen. So the first people that work with these antibodies expected the affinity was also worse, yeah, because they said, okay, it's smaller, so probably doesn't interact so nicely. But indeed, if you observe the geometry of nanobody, you see that the CDR3 of nanobody extrude from the surface of the molecule. And so it goes towards cavities in the antigen. Yeah? So somehow it's almost complementary with respect to the conventional antibodies because it can recognize epitopes that otherwise are difficult to be recognized by conventional antibodies. Yeah? So it means that most of the cases, yeah, in the case of nanobodies, will be native structural epitope that will be recognized, but not linear, yeah, and vice versa. Yeah? So it means also that according to the application for your, in which you think to use your antibodies, yeah, it will be better to have one kind of antibody than another because nanobodies probably will not work in Western blot. Yeah? In most of the cases, they don't work. And in any other application which you have denaturated antigen, yeah? But can be very effective, yeah, when you have native antigen. Yeah? So this is one reason that has been exemplified by some experimental work. Yeah? So in this case, yeah, people analyzed where the different nanobodies developed against the one antigen bound that antigen. Yeah? And they found that most of them, yeah, so 5 on 6, they shared overlapping epitopes. So there is a strong bias towards some epitope. This is true for any kind of antibody. So even if you have a polyclonal serum, finally, probably the antibodies will, even if you have, I don't know, hundreds of different antibodies, probably recognize no more than 3 or 4 epitopes. Yeah? And the same also in this case. So 5 of them recognize the same epitope. Only 1 recognized a different region. Yeah? And with lower affinity, because it's not a typical configuration for it. And when we compare the region, so the epitope recognized by single domain antibodies, both from shark and from camelids, yeah, with respect to the epitope recognized by conventional antibodies, we see that they don't match. Yeah? So they recognize different things. So having a combination, for some application could be better. So let's say that you wish to block simultaneously more epitopes to avoid, resistance due to mutation and so on. Yeah, this provide a wider possibility of blocking for longer time the mutagenesis of the antigen. Yeah. So when we analyze then more in detail what happens when we compare the characteristic of the variable region of a conventional antibodies and of a nanobody, we see first that nanobodies have a CDR3 that is longer, yeah, than with respect to what is in conventional antibodies. But then there are also some other variation. Yeah, we said this protruding capacity, yeah, of so the orientation of this CD3 makes that the surface of contact, so the overall paratope surfaces, yeah, becomes larger with respect of the number of amino acids. And so finally, when you compare the paratope surface, they are comparable. And this explains why, indeed, in the reality, the affinity of conventional antibodies and VHH are very similar, closer. Yeah? There is also other things that have been not understood at the beginning when people start working with nanobodies, but now we have more and more structure of nanobodies, so people could analyze better what happened exactly. And they found that indeed the way of building the paratope is quite different. Yeah? So here is indicated, yeah, for the VH of nanobody and of conventional antibodies, yeah, the contribution of the different components. Yeah. So here, I mean, should be just the classical paratope formed only by the hyper region of the CDR1 to F3. And you see, most of the conventional antibodies consider contribution only from these 3 loops. But when we look at what happened in the nanobody, indeed we see that there are extremely different combinations, and also in the use of variation of the residue. So it means that nanobody have more plasticity in adapting to different kind of epitopes because they can bend especially the CDR3 and let it collaborating with residues coming from a framework, for instance, yeah, that are not so often included in the paratope of conventional antibodies. Yeah? Because conventional antibodies, it's because of the pairing of the 2 variable region is strongly oriented now, so you have really only the loops oriented outwards. But in the case of nanobodies, yeah, it can the CDR3 can be bent over some residues of the framework. Some framework can be turned and so on. We had also an example in which indeed the same, so one single nanobody can build 2 independent paratope, yeah, using different different amino acids on its surface. Yeah, so these make also quite interesting nanobodies because somehow they can cover epitopes that are not directly targetable by the geometrical possibility that conventional antibody can offer. Yeah? But then, yeah, when you are interested in research, you wish to somehow to improve, to move ahead, yeah, and see if there is something better or something that can widen the potential of a technology. Yeah? And here we can see that when we look at the different scaffolds we were just reported at the beginning, we see that using different strategy of hypermutation, we can create different kind of surfaces with completely different characteristics, yeah, that again, yeah, will be optimal for some surfaces of the antigen, so for some epitopes of the antigen with respect to the other. Yeah. So what does it mean? Well, it means that in many cases, what is important from a practical point of view is not that you just get a ligand with maybe high affinity for a specific antigen, but you wish to target a specific epitope, because that epitope has a specific function, or I don't know, it has no specific function, so you wish to target it without altering the physiology. So you can have different aims, so you need different reagents. Yeah? And maybe for getting this specificity of targeting, you need different structure, yeah? Because different geometry will enable to form different conformation of a paratopes and recognize different kind of epitopes. Yeah. So people work with different ligands. We know that nanobodies now one nanobody has been approved. Maybe something. Okay, has been approved from FDA, so it's on the market for direct activity, and then you can use also this different ligand as for drug delivery, so as just cargos for delivering something. And here we have more variability. You see how many different ligands are now in different phases of development, yeah, with the idea that you can deliver, I don't know, imaging elements of prodrugs and so on. Yeah? So it means that there is an interest that is related to this, how you say, functional reason. Yeah? Then, of course, there are also other reasons. It can be economic due to, for instance, protection of patents and so on. Yeah? We'll talk a little bit of these later on. But just I wish to introduce one specific ligand that attracted our attention because of some characteristic. Yeah? So this is the adiron, are called also affimers. They have two loops. Yeah? So somehow they are different from or expect they are different in terms of binding with respect of nanobodies. What are the advantages of this scaffold? Well, there is no cysteine, yeah, at least in the original molecule. So this can be an advantage for recombinant production in many cases. Yeah, a T-melding of 100 degrees. Yeah, so you see here. So it means that since it's extremely stable, yeah, you can expect that you can introduce a lot of mutation, yeah, without compromising the stability, and because of this, also the yields. This is at least the theory. We'll see then the reality is slightly different. Yeah, and you can express it at really huge amounts. I mean, the wild type of adherons, you get 200 milligram per liter without optimization. Yeah, so it's yeah. It's very good. one other thing that is quite nice, I would say, is that when you do a panning, yeah, with a library of this adiron, you can get binders for very different epitopes. Yeah? And so this is, for the reason I just explained, could be very interesting because somehow, yeah, there are extremely flexibility in the organization of these two loops. Yeah? So you can target very different epitopes. And this multiply the possibility of application. Yeah? So this is the amino acid distribution of the wild type. Here you see there is no cysteine. We created a library of adiron, but we said, okay, since anyway the T melting is so high, maybe we can risk a bit and use also cysteines. Why? Well, because cysteines, it's true that can create problems during expression, but it's also true that provide a huge differentiation in terms of structure possibility, because you can get one or also single or multiple disulfide bonds and yeah, bending and so on. Yeah? So we said, okay, let's see what happens. Yeah? And what we got, yeah, are ligands that are relatively good. Yeah? We tested on 4 or 5 different epitopes, yeah, antigens, and yeah, I mean, okay, it was fine. Results that you could say similar to what we got with our pre-immune library of nanobodies in terms of affinity, in terms of yields, probably a little bit better, but strongly dependent on indeed the tag that we added, yeah. And what we couldn't expect, was it is totally unexpected indeed, is there is no correlation, that indeed an inverse correlation with the T melting. Yeah? So we expect to have high yields with high T melting, low yields with low T melting. It's the opposite. So this we are not able to explain exactly why. What we observe is anyway that the library of affimers with respect to the original molecule has T melting. Usually they're extremely lower, still relatively good, yeah, sometimes very good, but is significantly lower than the original. Yeah? And this can be probably explained by the fact that the molecule is very small, yeah? So it's 90 residues. So if you change 18 residues, it's a lot. No, it's 20% of the overall dimension of your molecule. So it's really a major impact, no, on the of the structure of the molecule. But still, you see, you get relatively good yields. When we analyze this, well, you see, it depends a lot. I mean, in terms of affinity, what kind of antigen we used. Yeah. So we have this bycatcher, a very small peptide. So, okay, the affinity were relatively low in the range of 1 micromolar. But in the case of others, for like CRP, the affinity was in the units of nanomolar. Yeah? So, good for being a premium library. And we tested in biosensor, yeah? And they work pretty nice as capture units of immunosensor, yeah, and they work in most of the normal immunoreaction that you can use. Why we were interested in another kind of ligand? We say because of the geometry. This is one point. The other point is that there is a progressive competition towards small molecules, yeah? And this is mostly driven by the super resolution microscopy, yeah? Because people wish to have something that is closer and closer to the target, yeah? So even gaining 1 nanometer seems to be relevant. Yeah, I mean, people are interested in gaining anything you wish. So if you use a conventional antibody, since you need also secondary antibody, yeah, you arrive to 30 nanometers to the object. Yeah. If you use a nanobody, you arrive to 3 nanometers. With an affimer, probably you arrive to 1.5, 2 nanometers. Yeah. So it's the closest thing that you can get. So this is something that moves the research towards small molecule. Of course, this can be also an advantage when you think about penetration in tissues, in solid tumors, and so on. But it's one of the things, yeah. So having this on mind and thinking about this collaboration that was aimed to also to create a library that could be used by the colleague here in Istanbul, yeah, we thought, okay, we can try to optimize a synthetic library of nanobodies, but can we also check if there are alternatives? Maybe we didn't try in the past, but can be maybe even better than nanobodies. Yeah? So what we did, we made an analysis of small proteins from thermoresistant archaeobacteria. Yeah? Then we did an analysis of the structure and tried to understand how we could trim and mutate these scaffolds. And so they are just example of what we changed in this scaffold. So we used 4 of these proteins. This is just an example of a different combination of the tray. And then we tried to express them to see how they work. Yeah? And indeed, the results were not very promising, because when you move from the wild type, that usually it's in some cases relatively good, so to the mutants, the structure tend to collapse, even if the 3D models seems to predict something stable, but you never know. So you can get better results when you fuse this molecule to something like a GFP or another kind of large molecule, but you have a little bit of degradation due to the fusion partner. So we worked a little bit on this, tried different combination, but finally we decided it was too risky invest in a library with this kind of scaffold, because apparently it's not as good as nanobodies, not as good as adherons that could be the first alternative, we can say. And we arrived now to the idea of the libraries. So conventionally, we said people use the antigen to immunize an animal, and then they prepare the library, and then pan the library and start all the characterization procedure. This is long, it's expensive, but of course it has the advantage of using the somatic maturation inside the animal. So what you get is a library already strongly oriented toward one antigen. So you need also a smaller library. So if you wish a pre-immune library, you must have a larger one, as we showed in the first picture. So instead of having, I don't know, a dimension of 10 to the 6, 10 to the 7, you must have a dimension of 10 to the 9, 10 to the 10. So this is somehow people say. And once you have this premium library, okay, the first advantage is that you can use directly. You take the aliquot from the fridge and you start working. It's true that what you get in terms of affinity, usually it's worse. So we usually get maybe 10 to the 9. That it's good enough for several applications, but for others maybe not. So if you go to an immune library, you arrive also to 10 to the 11. So this is for sure an advantage towards the immunized library. But the premium library is more flexible in terms of panning. So it gives you more opportunities to playing around. So you can start with several maybe panning strategy, compare what happens, what you can get. So one of the advantages for sure is that you can pan against not immunogenic antigen or toxic antigen, because you work in vitro. So no problem with this. And you can try to get binders towards still unknown antigen that are relevant because, for instance, you can discriminate between one kind and another, so one cellular subpopulation with respect to another. So we talk about biomarker. We don't know what could be the biomarker for a specific disease. This is a way to look for not only the biomarker, but also the reagents that recognize that biomarker. Yeah? So we worked a lot in this sense with in cancer, using, so we say, normal cells and cancer cells, and trying to see what discriminate between the two. Yeah, so this is something. Then you don't use animals, so this also simplify the work, of course. Especially in EU, where the legislation is very strict. And then, I wish to come back to a problem that for people working in the field, it's always a subject of debate. Yeah. So, what is a library? When a library is good? Yeah. So we're talking about theoretical dimension, and this is the number of the clones that you get. Yeah. So you count the colonies. You say, okay, I have this dimension, but it doesn't say a lot about the library, because you can have a lot of constructs that are not functional. Yeah, so you have to remove them just because of mistake during the synthesis of the cloning and so on. Yeah, you can have then repeats. Yeah? So you have to remove also these. So finally, you get what could be the total diversity. This is an indication of the dimension of your library, but what would be really important to know is the functional diversity, not the total diversity. So how many of the different combinations that you have in your library can be expressed, are stable, soluble, and so on. This is something that is not possible to know a priori. So finally, we remain to an empirical analysis. So we test the library. If it works, it's fine. If it doesn't work, it is not. No? So you test with 5, 10 different antigens. You see what it gets, and you say, okay, this library works. Yeah, I can use probably also for other 100 different antigens. Yeah. So this is a little bit about what are the dimensions that I expect to start at the beginning in terms of dimension. So. Yeah, of course.
Humanization or antigenicity?
No, I mean, there is a company that works with that, and we know that the company works with most of the pharma companies, so the big pharma companies, but we have no information about immunogenicity and these kind of things. So we don't even know, I mean, probably so far there is no clinical trial, I guess, because I didn't see it. But the information is missing about the in vivo application, even if we know that the company works for in vivo application as well. But I don't know how they solve the problem, if there is a problem. Otherwise, could be that it's not immunogenic because maybe it's something that we eat with a salad, and I have no idea.
And if you were to humanize a protein like that or any of the other ones that you talk about, where would you start? I mean, it's easy with a nanobody because it's similar to a VHH, so
the problem of immunogenicity is related to fact that they are difficult to predict. No, so. Exactly, you expect that it's far away from what we have, and we could recognize it as a non-self. Yeah, but I don't know if they invest so much. Probably they found a solution for this, or they already tested, and it's not immunogenic for a reason that, because maybe it's just we are already used to be in contact with this molecule.
I was asking, like, if you want to humanize it.
Yeah, but maybe you don't need to humanize.
You don't need to, but if you were to, let's say you had to.
Okay, there is a phytocystatin. There are cystatin also from human and so on. They are not so far away indeed. maybe it's also this similarity that overcome the immunogenicity problem. The company indeed use two libraries. One is made with the then, yeah, I mean, probably several of you are quite familiar with the panning strategy. So the idea is that you have a collection, yeah, of elements that can be phages, can be also other structure. Yeah. What is important is that this structure, yeah, can physically link the phenotype, that in this case is a nanobody, or the other ligand, yeah, with the genotype that is conserved as DNA inside the structure itself. Yeah? So you have a collection, you challenge the collection with the antigen, you wash away what is not specific, you elute, usually by partial denaturation, the specific, you amplify, and then you analyze. Yeah? When you work with phage display, since phages are small, you cannot use sorting, no, in flow cytometer. So you must use some system that is anyway has a limited possibility of screening. And you screen just couple of hundred usually of clones. On a library, it has probably millions of clones. Yeah? So you analyze just a very small amount. And usually you already get enough good clones for working, but indeed you don't know if all the other clones are just repeats of the same you have, or could be the storage of better binders. Yeah? So because of this, we thought to improve the system, try to rationalize it. And first of all, we made a deep sequencing, just to understand if the theory of phage display corresponds to the reality. Why this? Because the theory says that every time that you amplify, yeah, you select for the best clones. Yeah? You eliminate the non-specific and you increase the number of the specific. Some results from the peptide research, yeah, indicated that there are some peptides that are not binders for any antigen, but are always amplified, just because they are very fast in the amplification. Yeah? So it is something that happens also with the nanobodies. And indeed, what we found is these results that show that the theory and the reality diverges. Yeah? So let's see what happens, yeah, when we sequence the round 2 of the panning. Yeah? We see that we have the best binder in terms of KD, yeah? That is low in number of sequence, and in the round 3 is amplified, but not in huge way. Yeah. Let's consider this one. So extremely worse in terms of affinity. Yeah. Same number in the round 2, but is better amplified. So this is totally unexpected. And if you see here, no, there are plenty of examples that do not run as it should be, yeah? So this is probably the only case that follows somehow the theory. So it means that the effect of secretion efficiency during phage amplification, yeah, favors some clones that are not good, but are just very efficient in amplifying. Yeah? Because of this, we reduced the panning to 2 rounds to avoid amplifying, indeed, not optimal binders. Yeah? And this was one of the conclusions to which we arrived. The other was to try to combine phage display with ease display. So ease display has another possibility with phage display, because you expose the binder on the surface. You normalize, with another tag, the density of the binders present on the surface. Then you use the label antigen. So you have two double labeling. And you wish to get the population that express both fluorescences. And you expect that here are the best binders. What is the advantage of EAST display? That you can pass the whole library. And so you can check all the millions of clones that you have, and then you sort just the best one. So what we thought is, okay, let's start. We make the first round of panning in phages, because you have a large library that's not compatible with what you can put in an EAST library. And then the output of the first round of panning, you transform in EAST, and then you make the sorting in EAST. So we did this in parallel with a normal panning with phage display. And again, what we found was not what you expected from theory and rational analysis. So what we found indeed is that in the phage display system, we got here just the best binder. So we got roughly 15 different sequences. And among these, E12 was the best in terms of affinity, in terms of also of yields. When we passed to the EAST, we got plenty of positive, but then they were just corresponding to two single sequences. You strongly select for two sequences that, for one reason or the other, are extremely well displayed on EAST. So these again say that according also to the system that you use for the panning, you can get different kind of results, because any organism used for display has preferences for some sequences with respect to the other. So it's a bias that you know, that you have to consider when maybe you go for selection. So sorry for somehow say more you know and more you make your life complicated. No, it's true, but it's good to know that maybe if something doesn't work in one system, you can switch in another. So this is the positive part of the story. Also consider when you work during the panning, especially if you have a premium library that is not biased, you can really tune a lot of factors in order to modify the output. Okay, the classical is decrease the antigen concentration every time and so on. You can use the preliminary heating, so you eliminate the binder that are not very stable. Why you do this? Maybe you eliminate binders that have very good affinity, for instance. But in terms of developability of the system, you don't wish then to have good binders that you are not able to express. And it's very difficult psychologically to eliminate a binder that was good, for instance, when you did the SPR, but you are not able to express. If you do these kind of things, you eliminate everything that you already know will be not able to pass the downstream steps. You can work with washing stringency. This can be very interesting, especially if you have already a clear idea what is the application of your binders, and so you know that, for instance, in the final application, the binders will find, I don't know, specific pH values of detergent conditions, salt conditions, and so on, because you can introduce this during the panning. So select just the subpopulation of the binders that resist to the final condition that are supposed to be the experimental one. Then something that appear also quite interesting, it's tedious to do but useful, is to elute a progressive pH values, yeah? Because by such a way you get subpopulation of stronger and stronger binders somehow, yeah? This is not strictly direct connected to affinity, can be, but not only, yeah? But so it's important to have also this classification. And then also dilution by competition can be important, especially exactly if you wish to get binders for specific epitope, yeah? And I'll give you some example later on. So here just a schematic idea of what you can do, yeah, organizing the design of the panning, yeah, in order to get binders that makes different works, yeah, and that you can use in different applications. So you can organize in a way to get nanobodies that recognize several different kinds of similar proteins, similar antigen, yeah. So you need something that is a sort of generic binder for all a class of, I don't know, isoforms of an enzyme or what is, yeah? Or in contrast, you wish one that recognizes only one but not the other, no? So discriminate between this and all the other, yeah? Or just subgroups. Of course, you can play around. Or exactly that recognize specific epitopes, yeah? So you can try by panning design to organize the elution of binders that have this specific characteristic. And here I give you some examples. So here I show you the example of the panning that we do on cells or extracellular vesicles, yeah, because maybe it's simpler to understand graphically, yeah. So you start with a negative control, yeah. So you don't wish something that binds to this control, yeah. It can be healthy tissue, can be tissue 1 with respect to tissue B, and so on, yeah. Exactly. It depends on the design that you organize for your panning. So you challenge with your library, and you'll have two groups of phages, yeah. The one that will bind to this, and the one that not bind, yeah. So you eliminate this, and you keep this. And now you go on the target, and again you'll have binder and not binder. In this case, you keep the binders, you elute them, and then, for instance, you say, I wish something that binds to these cells, but also on these cells, and these and these. So you can try to get something that is useful for a class of cells, yeah? So this is one possibility. Another one that can be also quite interesting, no? Okay, you make the first negative depletion, yeah? But then what you wish, it's something that recognizes, yeah, the same, for instance, antigen, but murine and human, yeah? This is a classical example because you are interested in a human disease, but you have a mouse model. So you wish an antigen possibly that cross-react with both antigen and human and the murine, yeah? So you eliminate what recognize just the vector in this case, it is the egg cells, yeah? And then you use the cells that express, yeah, the receptor, the murine receptor, yeah? You take the positive, and now use the positive on the same, on the same kind of cells, but expressed in the human, yeah? And you'll have something that cross-react with these two and be negative with this, yeah? So you'll get something that recognize common shared epitopes between the two enzymes. So you can use then directly for comparing the data in the model and in your patients, for instance, yeah? You can try to get binders for a specific epitope, because you are not interested in just one binder for instance, in this case, HER2, but you wish one that is competitive with pertuzumab, yeah? So you start as before, but now, yeah, you are in this condition. Here, now, you bound, so you'll have for sure binders that recognize somewhere, no, the R2, but what you wish is something that recognizes just the epitope of pertuzumab. So you use pertuzumab to kick off that specific binder, not all the other, but only this, yeah? And now you have a binder that you can use for competing with pertuzumab, yeah? So an example that I find quite interesting is this here, yeah? So we work with FGF receptor 1. And when we did the first panning with normal elution by changing the pH, yeah, we got several different sequences, but all of the classes of a so-called C8. So they were overlapping for an epitope that was different from the epitope recognized by the natural ligand of the receptor, that is FGF2. Yeah. So we repeated the panning, because exactly when you work with premium libraries, it's not a problem changing conditions, start a new panning. And at this point, yeah, we've made a competitive elution with the natural ligand FGF2. Yeah? And we got two groups of binders, one that were overlapping of the two epitopes, and one that is specific just for the, we said, FGF2 epitope. Yeah? And, okay. Then we did the T elution, and so the pH elution, and we got again the binder of a C8 class. Okay? So this binder here, in terms of affinity, was not very good, but had this capacity of being competitive. So we wish to see how useful it could be. Yeah? And what we did was this experiment. So we express FGF receptor fused to GFP, yeah, and as you see, it's nicely displayed in the surface. After 24 hours, you have the same picture. If you add FGF2 plus heparin, after 24 hours, the receptor is totally internalized. If to these two components you use also the competitor of FGF2, yeah, you block the internalization. Yeah? So just to show that even if you don't select the best binder, yeah, but you select a binder with a specific function, yeah, then you can use to tune physiological condition. Yeah? So you get a binder that can be used for tuning some activities. Yeah? How you evaluate the clone, how much you can push the system. Yeah? It depends on the technique that you have available, yeah? Because exactly according also to the final application, you wish binders that maybe work in ELISA or work in SPR or work in flow cytometer. They are not usually the same. But any antibody has preferential for some application with respect to the other. Yeah? So what are the conditions which work? Yeah? How specific they must be. So here, just to show, we did a panning, yeah, against a kind of melanoma, yeah, and what we got were binders that were negative for the retinal cells. Okay, we wish this, and positive for cancer cells. But as you see here, yeah, blue and red correspond to a cell line, yeah, and the patient-derived xenograph from which this cell line has been recovered. So the PDX is polyclonal, and this is monoclonal. So this represents only one part of the diversity present in PDX. And as you see here, yeah, you can get totally different patterns. Yeah? So it means that recognize different antigen probably. Yeah? They are differently expressed here and here. Yeah? In one case, you have the red higher than the blue. In one case, you see it's only blue, the red is negative. In some cases, it's similar, so on. Yeah? So this means that you can already screen probably among all these positive clones for those that have or belongs to different functional classes, yeah, because probably they correspond to different antigens. And if you wish to do a very precise work of characterization, yeah, you can do this kind of work. So you have different cell lines, yeah, corresponding to different, in this case, tumors or control and so on. And then you see the intensity of each of these clones, yeah, according to the cell that you use. And you get, you see, groups of different patterns, yeah. So you can already say, okay, so this is a group that probably recognize, I don't know, one kind of epitope, another kind of epitope, and so on, yeah. So you can discriminate a bit and then try to make immunoprecipitation and the orphaning of the target, yeah. So this is a way for getting new biomarker paired to the reagent that can recognize this new biomarker, yeah. And yeah, you can, with a single panning, if you make a large-scale screening, yeah, already identify several potential epitopes that could be useful for characterizing one condition with respect to another. And with this, I finished, I guess. Yeah. Yes. So thank you for your attention, and if you have any question. Now, getting the structure of nanobodies relatively fast, so you can get high throughput models of the structure. So if you have a library of sequences, yeah, you could identify a library of structures, and then by docking, trying to find your match. So this is something we just started now to see, but indeed there are no available deep sequences of native library. We are just now trying to get one for ours, no? Try to see exactly what kind of information we get, and then how long does it take to make the corresponding structures. But for sure, I guess this will be one of the possibilities for the future. The other that is also promising, more innovative, if you wish. I just read recently an article. They built custom structure for any new epitope. So I cannot explain you because I didn't understand exactly how they do it, but the point is that you provide the structure of an epitope, and the system creates a corresponding surface. So it's not that it's a nanobody or what else. It's anytime a new scaffold somehow. So this would be maybe even the most advanced response, but I don't know how long does it take all the training for getting the information for making this work.
Actually, we are doing the second approach in our lab. We are trying to design the novel scaffold-free stable structures for specific epitopes. And the reason why I'm asking this question is the main problem that we are dealing with is flexibility of molecules. So nanobodies are also very flexible molecules and hard to dock. So the docking of rigid molecules are much easier, but docking of the flexible structures are a challenge. And finding proper tools for that kind of docking is challenging. And that's why I asked the question, if you are doing any kind of docking with
no, but
I would like to learn
there is maybe an article that could be interesting for you is about Lego bodies. from Rappaport, I think. So what they do is a very complex system indeed that binds in more points a nanobody. So they block a nanobody. So they use this for cryo-EM, no? But the point is that they take like a ganache, the nanobody, yeah? And so they reduce as much as possible the flexibility of the nanobody. This is for avoiding exactly flexibility that could create background in the picture of cryo-EM, but probably could
affect the affinity as well.
Sorry?
Could affect the affinity of binding.
They say that doesn't affect directly the affinity, but they block in one conformation. So the part that interacts should be flexible enough to interact, but all the rest must be blocked in that structure. Sorry? Lego bodies. Welcome.
But in any case, these in silico predictions are based on models. These models are not actually the real structures.
Yeah.
Although the AlphaFold and then others are working perfectly well, but we are still not so sure whether it is reflected in truth or not.
Yeah, I mean, the problem now it remains for the CDR3. Yeah, so for the rest, they are pretty good, but the CDR3 is very unpredictable in nanobodies, and these affect a lot the binding capacity. So yeah.
But before your question, can I ask something related to this scaffold? Well, as you mentioned, we are using these scaffolds, and what we have observed in MUM libraries, we used two lemmas, and we got two different sets of sequences from each lemma. Although conformationally they may have some similar structures, they bind more or less different parts of the protein, but the sequences were totally different in two lemmas. So, in terms of scaffolding and having libraries, do you think that there may be some critical differences in the libraries between different individual lemmas or camelids or others? Although they may bind similar targets, but the amino acid sequences may be different.
Yeah, so what we notice is that using our initial library, this is the native library, so we start from material from 20 different animals. Yeah? It seems to work better than a synthetic library we prepared with all, the rational idea that we developed over the years, because probably it provides two things that a synthetic library, even if larger, better organized, cannot do. And one is this variability in terms of geometry that can be. Yeah? And the other that is controlled by the evolution. So all the elements there somehow are functional, probably. No? In the synthetic library, probably you synthesize a lot of sequences that cannot fold correctly. Yeah? So this could be one of the reasons.
It's also true for in silico design of nanobodies as well?
Yes, but there are some smart development. So one library, it's very complex to do. This was done with a company and so on. But they identified three major scaffold folding structure for nanobodies. So they prepared three libraries indeed, and they fused these. So they have three different concepts of nanobodies mixed in one. So they reproduce more the naive. Yeah.
Yeah, to answer maybe your question, so there's a lot of nanobody sequences in the database because the PDB contains sequence and structure information. So using that you can figure out the percentages of naturally occurring amino acids in the CDR1, 2, and 3. And using that information, you can build your own library, but which we did in silico. And you know, when you screen in silico, you can get predictions of high affinity binders. But when you try to express this in bacteria, we tried it, you fail, because most likely these framework regions that have compensatory mutations that allow the mutation to be expressed in the biological setting are absent in the in silico library. So, so in our limited experience, we don't have these other compensatory mutations, and the protein just doesn't get expressed. So it's kind of useless to.
Yeah, when we do
The prediction of the model was perfect.
Yeah, prediction is excellent, but.
Yeah, when we do mutagenesis in order to optimize some characteristic, very often we arrive to the fact that then collapse somehow the mutants, no? So it's difficult. I mean, for having better affinity, usually we wish something that's more flexible, but it's more flexible, it's less stable, and it's difficult to get an equilibrium between the two. So from our experience, it's relatively simple to pass, from 1 micromolar to 20 nanomolar of affinity. But from 20 nanomolar to 1 nanomolar, it's extremely difficult. Yeah.
Using libraries.
Using in silico systems.
Any other question? Oh, yes, please.
So we always have trouble with the negative selection at the first step of our library in panic. So we usually use milk as the negative selection. What type of things do you use for the negative selection?
You mean when you're using a soluble antigen?
Yes, so like.
Yeah, I mean, apart plastic, I don't know if you use magnetic beads or these kind of things, you cannot do a lot because in that case you miss something that is very close and very diverse. As in the case of cells, no, it's a very nice system because you have the cell with all the epitopes present on the surface of the cells. No? So in that case, you can really discriminate the cells and the cells overexpressing one single protein. For a soluble antigen, this could be a problem. But reducing the number of panning could be one possibility, because otherwise longer goes with the rounds, and higher is the chances that you get these infectious sequences. So this is what we observe. Because when we started at that time, I mean, almost 20 years ago, people did 5 rounds of panning normal, no, usually. We passed first to 3 and then to 2. And that you can respect enough variability. And then you see that just decrease, but not towards the best one, just towards the most, robust somehow.
There's always a problem, and people use a panel of different proteins, DNA, to screen for specificity of their antibodies. Do you use anything like that to measure or see the specificity of antibodies?
Well, it depends on the kind of product, no? Because exactly, this is something that you can use during already the panning. If you know that you wish to avoid cross-reactivity with something, you can make the negative selection with this. Yeah. So this is always the best. I mean, if you already know that you wish to avoid some cross-reactivity and so on, I would use this during the negative selection before starting the panning. Because later on, it's always more complicated. You have more extremely higher number of clones, becomes a lot of work. But if you can eliminate at the beginning, because you already know that you wish to avoid, I don't know, some blood proteins or this kind of thing, because this is your final aim. So the general advice is always not starting the panning just to find binders, but having exactly in mind what is the final application, what you wish to get, what you wish to avoid, and then organize the design of the panning according to this. And trying to get rid of most of the unspecific or unwished during the initial depletion.
Small question. According to our experience, if your CDR net charge, especially CDR3's net charge, is over +2, it is mostly non-specific. This is our experience. Just we always check the net charge of our CDRs just to make sure we are not going in the wrong direction.
I don't know, in the sense that there are many people that try to infer some general rules from the sequences, and finally, when you analyze many, it's not possible really to get clear trends. No, I'll show, for instance, one example tomorrow. We used this predictor for stability. It's called PROS. You can apply to any kind of protein. Now we try to see if it works with a nanobody. In that case, for that specific nanobody, we got a hotspot in the CDR1. And also for a CDR1, the people said, yeah, you must have, I don't know, that residue here and there. It's true that in some cases it is a little bit stretched, no, the CDR1, but it depends exactly what are the other residues around. No? In that case, because we had this impression that was somehow blocked, we added just a glycine in between and became stable and didn't lose the functionality. No? But according to people said in the years, what are the rules for making this and that, it doesn't follow what people try to identify. I guess that in any case, it's better to trust information that comes from structural work than from sequence, simple sequence, because especially CDR3 very often interacts with framework residues. Yeah, so sometimes you must have something that's unstable there because stabilize some unstable patches in other regions. So.
Yeah. So I mean, following up on that, our experience also, it's very difficult to do negative selection. In your case where you have the 293 cells as the negative selector, and then the 293 cells plus your antigen as the positive selector, is there a ballpark sort of figure? How many fold more of the negative selecting cells?
Well, when you use cells, we use twice 10 millions of negative for the depletion, and for a positive you need even 500,000. You don't need a lot.
So we do that, and then even that, it's, with the yeast display, you get most of the binders you get are against the non-specific antigens on the surface.
With yeast, I don't know if it's sticky something or.
Maybe as a last question, the genic potential is quite critical for light chain of immunoglobulins. Have you heard anything about the immunogenic potential of nanobodies, VHH antibodies? Considering the scaffold of the protein, they have some potential.
I know that,
what is the nature of the main part, let's say, make fibrils of amyloid in a way?
I never heard of this. What we noticed is that the aggregation that we have is mostly colloidal, yeah? Not related to the single molecule, but just the fact that there are patches of interaction for others. And so this is against, we said, amylogenic. No, yeah.
Thank you very much.
Welcome.
It will be a pleasure to listen to
I
tomorrow
have
as well.
to remove.
So our next speaker is Sibel